Hello, my name is Catherine Leon, and today I will be talking about the research I have done at Montclair State University as an undergraduate biochemistry student.
Impact of S220A and E168A mutations on Mycobacterium tuberculosis Indole-3-Glycerol Phosphate Synthase, IGPS catalysis. This enzyme IGPS is required for the survival of Mycobacterium tuberculosis. Our goal is to investigate the substrate binding and the catalytic mechanism of IGPS to facilitate the discovery of inhibitors. To do this, single-point mutations were done to amino acids at the active site to investigate their roles in catalysis. The study of these two mutations, S220A and E168A, should allow us to elucidate the mechanism of this enzyme. The IGPS enzyme catalyzes the conversion of CdRP to IgP in the tryptophan biosynthetic pathway in bacterial organisms. On top, we see the overall reaction, and here we see a proposed mechanism. The enzyme facilitates a ring closure, followed by a decarboxylation, and finally a dehydration. This reaction is essential for bacterial life, and the enzyme catalyzing it, IGPS, is absent in humans. For these reasons, this enzyme represents a potential target for antibacterial drug development against pathogenic bacteria. To study how this enzyme interacts with ligands and catalyzes their reaction, a plasmid coding for the enzyme was transformed into E. coli cells. Kanamycin resistance was used for cell selection, and then cultures were grown to an ideal optical density. At this point, the cells were induced for enzyme production. The enzyme was purified via a histidine tag and a nickel column. Once the protein is purified, we visualize the enzyme and its purity on an SDS page gel. To make the mutants, single-point mutations were introduced into the plasmids and confirmed through Sanger sequencing. The substrate, CdRP, was synthesized by Dr. Konas and his research group at our Montclair labs. So, are the amino acids serine and glutamic acid essential for catalysis at positions 220 and 168 respectively? Each mutation was done individually, and the enzymes were tested using the wildtype enzyme for comparison. This image shows part of the crystal structure of Mycobacterium tuberculosis IGPS and its product IgP. Here we see that glutamic acid and serine are both in the active site, and they're both within proximity of the product. Although the enzyme and product and enzyme substrate interactions are not exactly the same, because these two amino acids are in proximity of the product, we suspect that mutating them to alanine, as seen here, would greatly decrease their activity. Before we can study the mutations, we have to study the wild-type enzyme. All enzyme kinetics were measured in a cuvette using fluorescence, with an excitation wavelength of 280 nanometers and an emission wavelength of 340 nanometers. In this case, we are measuring the appearance of product. On the bottom, we see a substrate-only control. These two reactions that are actually superimposed do not contain any enzyme. So over time, no product will appear. When we add the wild type enzyme, over time, we see an increase in signal, which corresponds to the product formation. O'Shane Thomas, a member of a research group, characterized the wild type activity by calculating its kcat. The kcat represents the number of substrate molecules each enzyme converts to product per unit time. This enzyme, IGPS wild type, was found to have a kcat of 0.6 plus or minus 0.17 per second. Using this kcat as reference, I attempted to characterize the mutations in the same manner. This figure shows a couple of the many kinetic experiments that have been done on the S220A mutation. The serine at the 220th position mutated to alanine. We can also see that here, where the hydroxyl group is basically changed to a methyl group. Here in figure 2, we see the substrate only control as the gray line. We also have the wild type control in blue, and I would just like to note that this is done at 100 nanomolar. Both of these controls were actually seen in the previous slide as well. Since mutating serine to alanine, it's a drastic mutation, I expected to see decreased activity in the mutated enzyme relative to the wild type enzyme. Here we see two kinetic runs for the mutant S220A at different concentrations. When the mutated enzyme is added to the reaction, there is no significant positive slope in either run. This is a trend that is seen when various enzyme and substrate batches are measured. I would also like to note that the mutant kinetic runs begin at a higher value on the y-axis at time 0. This is due to the increased concentration of enzyme in cuvette. For the wild type, we only require 100 nanomolar. However, because we weren't seeing any activity with this mutant, we increased the concentration to 3 micromolar in yellow and 4 micromolar in orange. Now, looking at the other mutation, E168A, we changed the glutamic acid at the 168th position to alanine. Again, this is a drastic change that we can see right here. So we expected to see a decreased activity in comparison to the wild type enzyme. Figure 3 shows some of the kinetic experiments performed on the E168A mutant. Using the same controls, the blue line represents the wild type enzyme, while the gray line represents the CdRP only control. We attempted to characterize this mutation. We see 3 different runs for mutant E168A, and all 3 of these runs show no significant increase in slope. Although there have been individual runs that are not shown here, where this mutation portrays small amounts of activity, meaning it had a bit of a positive slope. On average, most kinetic runs exhibit no positive slope, and that is what we see in this figure. This again has been observed while testing different enzyme and substrate batches. And once again here, we see that as more enzyme is added to the cuvette, the signal at time 0 increases. So the dark blue line is wild type IGPS at 100 nanomole. Then we increase a little bit when we add 1 micromole of the mutant. This is 2 micromoles and this is 3 micromoles. Serine and glutamic acid at their respective positions are definitely important for enzyme activity. When these amino acids are mutated to alanine, all enzyme activity is lost. This suggests that these amino acids could be involved in substrate binding, catalysis, and/or active site stabilization. Understanding the roles that these amino acids play will contribute to future drug discovery efforts. Moving forward, we would like to make more subtle mutations at these positions, and that should allow us to characterize these mutants. I would like to thank our NIH grant. I would also like to thank O'Shane Thomas, Thomas Candela, and Melanie Silva for their hard work in this project. Also, I would like to acknowledge Dr. Gubler, Dr. Konas, and Dr. Goody for putting this project together and working towards its success.
Thank you. I will be checking the comments to answer any questions, and I hope everybody stays safe.